Hi everyone. In this session, I'm going to talk about blood with special focus on plasma and erythrocytes in terms of composition and function. The objectives of this session are to list the composition and functions of blood in general, then to talk about plasma proteins, their functions and normal values, then the erythrocytes or red blood cells, their normal shape, their normal count, and the function of red blood cells. Also, I'm going to talk about hemoglobin in terms of structure, normal value of hemoglobin, the different types of hemoglobin that are present in our blood, and the different forms of hemoglobin, whether they are bound to oxygen or carrying oxygen, carbon dioxide, or other forms of hemoglobin. I'm also going to talk about the genetic abnormalities of hemoglobin and the process of erythropoiesis, which is the production of red blood cells, and how this process is regulated. And then I will talk about the destruction of aged or worn-out erythrocytes and how hemoglobin is catabolized. And finally, I will talk about jaundice, which is related to hemoglobin metabolism and the different causes of jaundice. The blood is a dynamic and complex living tissue carrying out four major functions, and these are: number one, transport of different substances like oxygen, carbon dioxide, nutrients, waste product between the alimentary canal and peripheral tissues, lung peripheral tissues, and from peripheral tissues back to the lung and the kidneys. It is also involved in the process of hemostasis, where it participates or contributes to the arrest of bleeding. It is also important for the maintenance of stable internal environments, like maintenance of body temperature, blood pressure, and so on. And also it aids in resisting infection or diseases, as white blood cells that are one of the components of blood are part of the immune system, or they contribute to both innate and acquired immune responses. The blood is a specialized connective tissue containing formed elements like red blood cells, white blood cells, and platelets, and intercellular matter or extracellular fluid that is contained inside the blood vessels and heart, or inside the cardiovascular system, and this intercellular fluid is also called plasma. If we take a blood sample and placed in a test tube, and this test tube is exposed to centrifugation, the blood will separate into three different components. One component, which is the plasma, that accounts for 55% of the whole blood volume. It is the liquid or the watery part of the blood that is composed mainly of water plus plasma proteins and other chemicals, ions and chemicals. Then we have the erythrocytes, and these represent 45% of the whole blood volume, and plasma and erythrocytes are separated by a thin buffy coat that represent less than 1% of the whole blood volume, and it is composed, or it contains, leukocytes and platelets, the white blood cells and the platelets. Now let's go and talk about the composition of the blood. Blood average volume is different between male and female. In male it is about 5.5 liters, while in female it is about 5 liters. It is a bit less because female usually they have on average less body mass compared to male. The composition of blood is mainly plasma, 3 liters, which is a little bit more than 50% of the blood volume. Then we have the formed elements, or the blood cells, and these are mainly the red blood cells, also called erythrocytes. Erythro means red, so we have the red cells. In men, they represent 42 to 52% of the total blood volume, while in female, the red blood cells represent 37 to 47% of the total blood volume. It is less in female due to the monthly loss of blood during the menstrual cycle. Then we have the white blood cells, or WBCs, also called the leukocytes. And finally, we have the smallest blood cells. These are the platelets, also called thrombocytes, because these are the cells that contribute to thrombus formation. Okay. The plasma, which is an important component of blood, is composed mainly of water. 90% of the plasma is water, and another important component of the plasma are proteins, and we call them the plasma proteins, and they represent 6 to 8% of the plasma volume. Then we have about 1% of different electrolytes, mainly in the form of sodium and chloride, because these are the main ions that are present in the extracellular fluid, and plasma is one of the extracellular fluids. So these electrolytes or the plasma contains relatively high concentrations of sodium and chloride and relatively low concentrations of hydrogen, bicarbonate, potassium, and calcium. Then we have other molecules, either nutrients or waste products in the form of glucose, amino acids, lipids, vitamins, bilirubin, urea, and creatinine. In terms of plasma proteins, we have about 1,400 different plasma proteins that were identified so far in the human being, and these amount to a total of about 6 to 8 grams of proteins per deciliter of plasma. These proteins play a very important role in our body as they have enzymatic function, or they can act as hormones, antibodies, or transporters. So we can see those different functions that play very crucial role in maintaining homeostasis. The plasma proteins contribute to plasma osmolarity, as most of the plasma proteins have large size, so they cannot cross the capillary membrane. They are retained inside the lumen of the cardiovascular system, and we know very well that plasma proteins or proteins in general can exert an osmotic pressure. That's why plasma proteins contribute to the plasma colloid oncotic pressure. And also proteins have the ability to gain and lose hydrogen, so they can act as buffers and play a very important role in the acid-base balance. Plasma proteins, when we have nutritional deprivation, they can serve as a source of energy. Under limiting conditions such as malnutrition, they will be broken down into amino acids, and the amino acids will be used as a source for energy when we are deprived of nutrients. How many types of plasma proteins? We have at least 3 main types of plasma proteins. The most abundant plasma protein is albumin. 60% of all plasma proteins are albumins, and this is about 3.5 to 5.5 gram per deciliter of plasma. Albumins are produced by the liver, and they contribute to the plasma osmotic pressure, the transport of fatty acids, hormones, drugs, and other substances in the blood. In addition to their important role as contributor to the plasma colloid oncotic pressure, they act as transporter for the water-insoluble molecules or the fat-soluble molecules like fatty acids, the lipid-soluble hormones, some lipid-soluble drugs, and other substances in the blood. The second type of plasma proteins are the globulin, and they account for 36% of the plasma proteins, which is about 2.3 to 3.5 gram per deciliter of plasma. We have 3 different types. We have the alpha and beta globulins. These are produced by the liver and function as transporters and substrates. And we have the gamma globulins, and these are the only plasma proteins that are produced by the lymphoid tissue, by the plasma cells. The activated beta cells or B lymphocytes, when they are activated during the immune response, they transform into plasma cells that produce antibodies, and these antibodies are gamma globulins. These antibodies are necessary for the immune defense. Finally, we have another important plasma protein that is very important for the final step of hemostasis, where we stop bleeding, and this is fibrinogen. It is inactive form of fibrin. It accounts for 4% of the plasma proteins. It is a large molecule produced by the liver, and it is converted to fibrin, which is insoluble, and this fibrin will be part or will contribute to the formation of the blood clots. Now let's talk about the red blood cells, the other important or the second important part of this session. Red blood cells, as I said, they are also called erythrocytes. Erythro means red and cytes means cells, so these are red cells. How do they look like? They are flat, disc-shaped, indented in the middle. So they are biconcave. They have a diameter of about 7.5 to 8 micrometer, and they have a thickness at the thickest part of 2 micrometer. So they are flat, discoid, biconcave cells. Is it important to have this shape for red blood cells? Yes, of course. This shape is very important for their function. They have a very strong, flexible plasma membrane, and this flexibility of the plasma membrane is very important to allow the red blood cells to squeeze or deform themselves when they pass through the very narrow capillaries that have a diameter of 2 to 3 micrometer. And as the red blood cells advance in age, the flexibility is lost, so their cell membrane becomes stiff, and that's why they become worn out, and the phagocytosis will remove them from the circulation, as we'll see later on. The plasma membrane of red blood cells also has an important feature, as it carries or contains glycolipids. And these glycolipids are responsible for the ABO and the RH blood groups that we'll see in one of the coming sessions. So these glycolipids determine our blood type. One important feature of the red blood cells, they are non-nucleated or anucleated, so they have no nucleus, and since they have no nucleus, they cannot reproduce because they lack the genetic material that is important for the mitosis. What are the major functions of red blood cells? The most important function of red blood cells is to transport oxygen and carbon dioxide. Red blood cells transport about 98.5% of oxygen that is carried in the blood from the lungs to the peripheral tissues, and also red blood cells carry back carbon dioxide from the peripheral tissues to the lung, but only 20 to 23% of carbon dioxide. So it is mainly a transporter of oxygen and to a lesser extent a transporter of carbon dioxide, because most of the carbon dioxide is transported in the plasma in the form of bicarbonate after being converted to bicarbonate inside the red blood cells. The lifespan of red blood cells is about 100 to 120 days. So on average it is about 4 months. Then the worn out stiff red blood cells are removed by macrophages present in the spleen, liver, and the red bone marrow, but they are mainly removed by macrophages present in the spleen. Men on average have about 5.4 million red blood cells per microliter of blood, and women have less because, as I said earlier, due to the monthly loss of blood during the menstrual cycle. So females or women usually have about 4.8 million red blood cells per microliter of blood. To have a stable or fixed number of red blood cells in our blood, the rate of production of red blood cells should be equal to the rate of destruction. So on average, we produce, or our red bone marrow produces about 2 million new red blood cells per second, and our macrophages in the spleen, liver, and red bone marrow remove about 2 million worn out red blood cells per second. That's why we have, or normally we have, stable or fixed number of red blood cells. The red blood cells have another important feature. They have no mitochondria. And we know that mitochondria are responsible for the aerobic metabolism. Since they have no mitochondria, red blood cells do not use any of the oxygen that they carry or transport, because they have glycolytic enzymes to generate ATP anaerobically without the need of oxygen. So they transport oxygen without using that oxygen. The red blood cells are red because they contain oxygen-carrying protein, which is called hemoglobin, that was synthesized or produced inside these cells before the loss of the nucleus, because we know that genetic material is very important for the production of proteins. Inside one red blood cell, we have about 250 million hemoglobin molecules per cell. So one cell or one red blood cell contains about 250 million hemoglobin molecules. The biconcave shape of the red blood cells provides a larger surface area. If they were spherical with the same diameter, they will have less surface area. So this biconcave shape will provide them with a larger surface area, and this is very important for the exchange of oxygen between the alveoli and hemoglobin in the red blood cells, and also in the peripheral tissue between hemoglobin and the peripheral tissue cells. And the flat shape or the thinness of the red blood cells give a shorter distance for oxygen to travel in and out of the cell. So oxygen can move quickly in and out of the cell because it has only short distance to travel across the cytoplasm and then the plasma membrane. What is the composition of hemoglobin molecule? The hemoglobin molecule, as you can see here, it is composed of 4 polypeptide chains, and these 4 polypeptide chains are 2 alpha chains, here the light blue ones, and 2 beta chains, the darker blue polypeptide chains. And each chain has a heme ring, this yellow heme ring. And as you notice, in the center of the heme ring, there is iron in the ferrous state. A ring-like non-protein pigment, this one, called the heme, that is bound to each of the 4 polypeptide chains. So one hemoglobin molecule has 4 polypeptide chains, and each chain has 1 heme, so totally we have 4 heme rings or pigments per molecule of hemoglobin. And each heme has iron in its center. At the center of each heme ring, and there is an ion of iron in the ferrous form that can combine reversibly with oxygen. And one ion can bind only 1 oxygen. And I want to attract your attention here that iron should be in the ferrous form to bind to oxygen. If it is in the ferric form, which means 3+, it cannot bind oxygen. It should be in the ferrous form so it can reversibly bind with 1 oxygen molecule. Since we have 4 heme rings per molecule, 1 hemoglobin molecule can carry a maximum of 4 oxygen molecules. So this is the maximal capacity of 1 hemoglobin molecule, is to carry 4 oxygen molecules. So each hemoglobin molecule can bind 4 oxygen molecules. In the lung, oxygen is picked up and binds to the ferrous ion. Then it is carried and released to the peripheral tissues. Oxygenated hemoglobin that is carrying oxygen is bright red, while deoxygenated hemoglobin that has released its oxygen to the peripheral tissue, has a dark red color. So there is a color change in the hemoglobin molecule based on how many oxygens are carried, or whether oxygen is carried by that hemoglobin or not. Hemoglobin also, as I said earlier, transports about 20 to 23% of the total carbon dioxide in the blood, which is picked up in the peripheral tissues as a product of metabolism and then released in the lungs. Please notice that carbon dioxide combines with the amino acids of the hemoglobin molecule. So carbon dioxide does not bind to the ferrous iron. It binds to amino acids that are present in the polypeptide, because we know that polypeptides are composed of amino acids. So there is no competition between carbon dioxide and oxygen for the binding site on the hemoglobin molecule. The normal range for hemoglobin level is about 14 to 18 grams per deciliter of blood in men and 12 to 16 grams per deciliter in women. Again, it is less in women due to the monthly blood loss during the menstrual cycle. How many types of normal hemoglobin do we have? And again, I'm stressing on the word normal. We have 3 different types of normal hemoglobin that can be found in adults. And one of them is mainly found in fetus or in the newborn. And then as we grow up, it decreases to very low level. What are these 3 types? We have hemoglobin A, or adult hemoglobin. That's why it's called A, based on adults. It represents about 95 to 98%, so between 95 to 98% of the total hemoglobin that is found in adults. So this is the main form of hemoglobin that is present in our red blood cells as adults. It contains 2-alpha, or the hemoglobin molecule is composed of 2 alpha chains and 2 beta chains, as I explained in one of the previous slides. So that was about adult hemoglobin. Then we have another form of hemoglobin, which is called hemoglobin A2. It was discovered after A, and they didn't want to label hemoglobin A as hemoglobin A1. They just labeled this as hemoglobin A2. So this is the second type of adult hemoglobin. It represents 2 to 3% of hemoglobin that is found in the red blood cells of adult human being. It has 2 alpha chains and 2 delta chains. So the difference is having delta chains that are different from beta chains in the sequence of amino acids that are present in the polypeptide chain. Then we have the hemoglobin F, which is fetal hemoglobin. This is the main type of hemoglobin that is found in fetus or then in the newborn, then gradually decreases in level till the age of 2 years. So it represents about 1 to 2% of hemoglobin that is found in the red blood cells of adults. It has 2 alpha chains and 2 gamma chains. So the difference is in the gamma chains, not the alpha. Alpha is present in all types of hemoglobin. It is the primary hemoglobin produced by the fetus during pregnancy, and its production usually falls shortly after birth and reaches adult level within 1 to 2 years. So from 90% or more than 90% of hemoglobin in the fetus and newborn, it will reach only 1 to 2% after 2 years, when we are 2 years old. Now, what are the different forms or derivatives of hemoglobin? Assume we have hemoglobin A, and what are the forms of hemoglobin A? We have something called oxyhemoglobin, and this is hemoglobin with an oxygen attached to the ferrous ion or the ferrous iron ion. It is bright red in color, as I said earlier. We have the oxyhemoglobin. It is hemoglobin without oxygen. So hemoglobin that releases all the oxygen to the peripheral tissue, it becomes deoxyhemoglobin, and it is dark red in color. Then we have carbaminohemoglobin, hemoglobin with carbon dioxide group or carbon dioxide gas attached to the amino group of the globin molecule. It is dark red in color. Again, I repeat, carbon dioxide binds to amino acids on the polypeptide chains, not to the ferrous ion that is present in the heme ring. So there is no competition between carbon dioxide and oxygen for the binding site on hemoglobin molecule. We have another form of hemoglobin which is abnormal, which is pathological, and this is really serious. It is called carboxyhemoglobin. It is hemoglobin with carbon monoxide. When carbon dioxide or when carbon is incompletely burnt, when we have shortage of oxygen, this will produce carbon monoxide. Carbon monoxide is dangerous because it binds to the ferrous ion in the heme ring. Carbon monoxide binds to the ferrous about 200 to 300 times more tightly than oxygen. So the affinity of carbon dioxide for iron in the heme ring is 200 to 300 times higher than oxygen. So it competes with oxygen and it displaces oxygen. So hemoglobin cannot carry oxygen from the lungs to the peripheral tissue. So oxygen cannot displace carbon dioxide due to this high or tight binding between carbon dioxide and iron, and this will lead to RBCs that are not able to transport oxygen, and this condition can lead to death. We have another form of hemoglobin called methemoglobin. It is formed by the oxidation of heme ferrous to ferric iron. As I said earlier, hemoglobin with ferric iron cannot bind oxygen. It cannot bind oxygen. Normally we have about 0.5 or about 0.5 to 1% of our total hemoglobin in the methemoglobin form. This is due to the oxidative stress that we are exposed to on a daily basis. But luckily we have very important enzyme that will convert back the ferric iron in the heme ring to ferrous iron, and this enzyme is called methemoglobin reductase. And this enzyme will keep the methemoglobin at very low level, which is 0.5 to 1%. We have another form of hemoglobin called glycated hemoglobin, hemoglobin A1C. This hemoglobin is normally present, or this form of hemoglobin is normally present in our red blood cells. Why it is present? Because glucose that we get from after the digestion and absorption of carbohydrates can bind to any protein in the body, including hemoglobin, without the need of any enzymes. So hemoglobin, whose globin chains possesses non-enzymatically attached glucose molecules. And as I said, it is normal, it can happen due to the glucose that is present in the plasma. It will enter the red blood cells and it will bind to the globin and produce the glycated hemoglobin. Usually, healthy person has values of hemoglobin A1C less than 5.7% of their total hemoglobin. So if we are not diabetic, our glycated hemoglobin will be less than 5.7%. Diabetic people that are not taking any medication or they are not compliant with their medication, they have much higher values. They have more than 6.5% of their hemoglobin in the form of glycated hemoglobin. Any values between 5.7% and 6.5%, this is a condition called prediabetes. So this is present in pre-diabetic people. So it is an indicator that this person, if not treated or if he doesn't change his lifestyle, most likely he will develop diabetes later on. Now, what are the genetic abnormalities of hemoglobin? we have different abnormalities that result from different causes. One of them is the synthesis of abnormal hemoglobin, like hemoglobin S in sickle cell disease, where glutamic acid in the beta chain is replaced with valine. The glutamic acid, which is an amino acid, in the beta chain is replaced with valine, and this will produce abnormal beta chain, and the abnormal beta chain, two abnormal beta chains with two normal alpha chains will produce hemoglobin S. And the problem with hemoglobin S, if we have a condition with low oxygen in the blood, the hemoglobin will undergo conformational change, and this will lead to the sickling of the red blood cells. They will become crescent-shaped, not flexible, so they will aggregate in small blood vessels. They will undergo hemolysis, they will release their content, and this is a big problem. Also, we lose our oxygen carrying capacity because hemoglobin outside red blood cells cannot transport oxygen. Free hemoglobin is not able or cannot transport oxygen from the lungs to the peripheral tissue. It should be contained inside intact red blood cells. We have another abnormality where we have reduced rate of production or synthesis of normal alpha or beta chains. For example, there is a condition called alpha thalassemia where there is reduced production or synthesis of alpha chains. So there is excess production of beta chains in the adults and excess production of gamma chains in the newborn, because for fetal hemoglobin, the beta chain is replaced with gamma chain. So this produces hemoglobin H, so hemoglobin with 4 beta chains rather than 2 alpha and beta. So we don't have the adult hemoglobin. We have hemoglobin H in the adults, and we have another hemoglobin in infants and newborn called hemoglobin Barts, because the absence of alpha chain will be replaced by gamma chains, so we have hemoglobin molecules with 4 gamma chains, and both types of hemoglobin are abnormal. Alpha thalassemia is very common in the Far East. We have another form of thalassemia called the beta thalassemia, where there is decreased synthesis of the beta chain, and this will lead to high level of hemoglobin F or theta hemoglobin in the adult, with normal levels of hemoglobin A2, because it's composed of 2 alpha chain and 2 delta chains, and these 2 are not affected in beta thalassemia. This beta thalassemia, or this condition, is more common in the Mediterranean region. Now let's talk about the process of red blood cell production that happens in the red bone marrow, and the process is called erythropoiesis. It starts in the red bone marrow with a precursor cell called pro-erythroblast. So this is the precursor cell. The pro-erythroblast will undergo into several steps of division and maturation, and finally at one stage the nucleus will be ejected and will have a new cell that is called reticulocyte. The pro-erythroblast divides several times, producing cells that begin to synthesize hemoglobin. Cell near the end of development, after hemoglobin was produced, will eject the nuclei, and they will become cells that are called reticulocytes. These reticulocytes now can be released from the red bone marrow into the circulation. Reticulocytes leave the bone marrow into the circulation, where they circulate for 1 to 2 days until they reach full maturity and become mature red blood cells. Reticulocytes develop into mature red blood cells within 1 to 2 days after their release from the red bone marrow. Normally, erythropoiesis and red blood cell destruction occur at roughly the same pace or rate. As I said earlier, to have a fixed or stable number of red blood cells, the amount of red blood cell production or the rate of red blood cell production should be equal to the rate of red blood cell destruction that happens in the spleen, liver, and also to lesser extent in the red bone marrow. Erythropoiesis is controlled by a negative feedback system that is very important controller of the process. And this is done as follows. If we have a low oxygen carrying capacity, or if we have anemia, oxygen delivery to the kidneys will become less. Special cells in the kidneys, we call them the peritubular cells that are present in the renal cortex, will sense this low oxygen level and will produce the hormone called erythropoietin. So hormone called erythropoietin that is released by the peritubular cells of the kidney in response to hypoxia or low oxygen level. This hormone will be released into blood. It will circulate to the red bone marrow and will stimulate the proerythroblasts to divide and go into several stages of development to give us reticulocytes that will be released into the blood. This will mature into red blood cells, and now we have more red blood cells that can carry enough amount of oxygen to the kidneys, and this will stop the release of erythropoietin. So hypoxia, which is cellular oxygen deficiency, like living at high altitude, anemia or reduced blood flow to the tissues or to the kidney will stimulate the kidneys to release the hormone erythropoietin. Erythropoietin will be released in the blood, it will reach the red bone marrow, and there it will speed the development of proerythroblasts into reticulocytes that will be released into the circulation. The rate of erythropoiesis can be measured by the number of immature red blood cells or by measuring the reticulocyte count in the peripheral circulation. Normally, we have about 1 to 2% of red blood cells in the peripheral circulation in the form of reticulocytes. So if we count 100 red blood cells in the peripheral blood, 1 to 2 of these are reticulocytes, the immature form of red blood cells. If we have low reticulocyte count, less than 0.5%, this is an indication of low rate of erythropoiesis, while a high level of or elevated count or elevated reticulocyte count, more than 2%, indicates a high rate of erythropoiesis. So by measuring the reticulocyte count, we can have a rough idea about the level of erythropoiesis or the rate of erythropoiesis that happens in the red bone marrow. Any cell that is more immature than a reticulocyte, like proerythroblast, should never be seen in the circulation, and their presence indicates the blood tumor, which is called leukemia. So the most immature form of red blood cells that are present, that should be present in the circulation, are reticulocytes. Any other precursor cells that are preceding reticulocytes should never be seen in the circulation. Now, what are the abnormalities of erythropoiesis? Erythropoiesis can undergo into different abnormalities. One of them is anemia, which is very common. It is a condition of insufficient red blood cells or hemoglobin. So either we have low number of red blood cells, abnormal red blood cells, or a low hemoglobin or abnormal hemoglobin. So in terms of quality or quantity. It is most often the result of low iron intake, hemolysis, autoimmune disease, blood loss during surgery, trauma, or whatever, or lack of production in the bone marrow due to bone marrow failure for any reason. For example, radiotherapy as a side effect of any treatment, or sometimes the bone marrow will fail without any known reason. But the most common form of anemia that is present all over the world is the iron deficiency anemia, especially in female. The condition that is opposite to anemia is called polycythemia. Is a condition characterized by an excess number of circulating red blood cells and an elevated hematocrit or elevated number or count of red blood cells. It occurs in response to hypoxia, shots of erythropoietin, many athletes use injections of erythropoietin to increase their hemoglobin so they have more oxygen carrying capacity, so they are more competitive in any game and they can get a gold medal, but this is illegal. Also, COPD or chronic obstructive airway disease can lead to chronic hypoxia, and this will lead to polycythemia. Another condition that leads to relative or apparent polycythemia is dehydration. During dehydration, the volume of the plasma becomes relatively low compared to the red blood cell volume. So at this stage, we can, if we do the complete blood count or we do the centrifugation of blood sample, we can notice that there is a polycythemia. This is not a true polycythemia because when we treat the dehydration, it will disappear. Now let's talk about the life cycle of red blood cells. Red blood cells live only about 100 to 120 days, or on average 4 months. So what happens to the worn out red blood cells that lost the flexibility of their cell membrane, that are no more pliant? They cannot squeeze themselves easily in the small blood vessels, in the small capillaries. As we know, the red blood cells cannot synthesize new components. That's why they undergo aging, because they have no nucleus, they have no genetic material for producing their own components or their own new components after the ejection of the nucleus. Aged and ruptured red blood cells are removed from the circulation and destroyed by fixed phagocytic macrophages that are present mainly in the spleen, also the liver, and to a lesser extent in the red bone marrow, as I said earlier. The breakdown products of the content of the red blood cells, which is mainly hemoglobin, are recycled. So the globin, which is the polypeptide, or the globin and heme portions, are split apart. So the heme ring is split or separated from the polypeptide chain or from the globin. Globin is a polypeptide. It will be broken down into amino acids, and these amino acids can be reused for the production of proteins anywhere in the body, including new hemoglobin. Iron is removed from the heme ring, or from the center of the heme ring, and then it is reused for the production of new hemoglobin. The non-iron portion of heme will end as urobilin, which is yellow pigment in the urine, or stercobilin, which is brown pigment in feces. So the non-iron portion of heme is not recycled. It will be metabolized. It will go through several steps of metabolism and finally will reach urine in the form of urobilin. And this is partially responsible for the yellow color of the urine, or as stercobilin in feces, which also contributes to the brown pigment of the fecal material. So what happens exactly? This is a fixed tissue macrophage in the spleen, liver, or red bone marrow. The worn out red blood cells are engulfed by these macrophages, and then the cell membrane will be destroyed and the content will be released. Heme and globin will be separated from each other. Then the globin, which is the polypeptide chain, will be digested into amino acids that will lead the macrophage to be reused for the synthesis of different proteins in the body, including new hemoglobin. The heme ring, the iron will be removed from the center. It will be converted to ferric iron. Ferric iron will be released from the macrophage and will be carried by one of the plasma proteins called transferrin. Iron cannot be transferred or transported as free iron in the plasma. It should be carried by a plasma protein called transferrin. Transferrin will take the iron to the liver, where it can be stored in the liver bound to a storage protein called ferritin that is present in the cytoplasm. Or transferrin can transport iron to the bone marrow for the production of new hemoglobin. Either stored in the liver or directly carried to the bone marrow, the iron will be transported to the bone marrow bound or carried by transferrin. This iron will bind to globin, which is newly produced in the bone marrow in the presence of vitamin B12 and the most important hormone that stimulates erythropoiesis, which is erythropoietin, which is released in turn from the kidneys in response to the oxygen to produce new hemoglobin that will be packed in new red blood cells and released into the circulation. And these new red blood cells can survive for 120 days and the cycle will repeat again. Now, what about the non-iron part of the heme ring? It will be converted to biliverdin, which is a green pigment, and this biliverdin will be converted into bilirubin before it leaves the macrophage. Bilirubin will be released into the blood, carried bound to albumin, and delivered to the liver. The liver will process this bilirubin, and then it will be released with the bile into the biliary tree to the small intestine. Bilirubin will travel through the small intestine until it will reach the large intestine. There it will be converted to urobilinogen. Small amount of urobilinogen will be reabsorbed back into the blood and then converted in the liver into urobilin. Then urobilin will be released into urine, and this will give urine its yellow color. Most of the urobilinogen in the large intestine will be converted to stercobilin, and stercobilin will pass with the fecal material, and as I said, it will give the fecal material its brown color. So everything here, as you see, is recycled except the non-iron part of the heme ring. So let's summarize the steps that were in the previous slide. Macrophages will engulf the ruptured or worn out red blood cells. Globin and heme portions of hemoglobin are split apart. The globin is broken down into amino acids to be reused. Then iron is removed from the heme portion in the form of ferric iron and is carried by transferrin in the bloodstream, either to the liver or to the red bone marrow. In the liver, muscle fibers and macrophages of the spleen, the ferric iron detaches from the transferrin and is attached to the iron storing protein that is called ferritin. So be careful. The transport protein is called transferrin, which is plasma protein. The storage protein is called ferritin. So be careful and don't mix between them. Open release from the storage site or absorption from the GIT, iron binds to transferrin and it is carried to the red bone marrow, where red blood cell precursor take the iron and used for the production of new hemoglobin. Vitamin B12 is also needed for the synthesis of hemoglobin. Now, next, the erythropoiesis produces red blood cells which enter the circulation in the form of reticulocytes that will last for 1 to 2 days. When iron is removed from the heme, the non-iron portion of the heme ring is converted to biliverdin, which is green pigment, and then into bilirubin, which is a yellow-orange pigment. Bilirubin will enter the blood and is transported to the liver, as I said, bound to albumin. Bilirubin cannot be transported free in the blood. Within the liver, bilirubin is released by the liver cells into the bile, which passes into the small intestine and then finally the large intestine. In the large intestine, the microbiota or the good bacteria that are present in the large intestine will convert bilirubin into urobilinogen. Some of the urobilinogen will be absorbed back into the blood and converted in the liver to the yellow pigment called urobilin, which is yellow, to be excreted in the urine. Most urobilinogen is excreted in the fecal material in the form of brown pigment called stercobilin, which gives the fecal material its characteristic color. Now, what is jaundice? Let's talk about jaundice because abnormality in hemoglobin metabolism can or is one of the causes of jaundice. Jaundice refers to yellow discoloration of the skin, sclera, which is the whites of the eye, and the mucous membranes caused by hyperbilirubinemia, which means high bilirubin in the blood, higher than the normal range. What are the different causes? We can divide them based on their relation to the liver. So one type is called prehepatic jaundice, because here the cause of jaundice is something not related to the liver, like excess destruction of red blood cells in hemolysis. Excessive destruction of red blood cells will produce large amount of bilirubin, and this large amount of hemoglobin exceeds the maximum capacity of the liver to deal with that amount. And this happens, for example, in sickle cell disease. We have another type of jaundice called hepatic jaundice. This is due to the decreased or limited abilities of damaged hepatocytes or liver cells to deal with the normal amount of hemoglobin that is produced by the destruction of red blood cells, like in liver diseases, viral hepatitis, liver failure, and so on. Then we have a post-hepatic jaundice. This jaundice is usually caused by obstruction in the biliary tree, either by stone, by tumor, and other causes. So these are the different causes of jaundice in relation to the liver. We have prehepatic, hepatic, and post-hepatic. In our condition or in our session, jaundice is caused by excessive destruction or breakdown of red blood cells, like in hemolysis. Thank you very much for listening to this session, and I wish you all the best.